All right, let's take a deep dive into the world of oligonucleotides.
Oligonucleotides.
Yeah, they're these short chains of genetic material that are making waves in all sorts of fields, like
Forensics,
exactly, genetic testing, and
research into storing the world's data in DNA.
exactly, like, whoa, we're going to be using this awesome guide that you sent us to break down how they're made, what the different types are, and
all the amazing things scientists are doing with them.
yeah, all the amazing things scientists are doing with them, so.
It's pretty wild. What's really neat is how they're essentially like custom-made Lego blocks for DNA and RNA. This lets scientists work with genetic information at a level of detail that was just impossible a few decades ago.
Okay, so you've been studying these oligonucleotides for a while now.
Yeah, I've been fascinated by them for a long time.
Let's dive in then. The guide mentions that the name oligonucleotide actually tells us something important about their size. Oligo means few, right?
You got it. And when you see a term like 20-mer, it means the oligonucleotide has 20 of those building blocks, the nucleotides, all linked together. It's like specifying the exact number of pieces in a puzzle.
I see. So a 100-mer oligonucleotide would be 10 times longer than a 10-mer.
Exactly.
And they'd likely have totally different uses. That's a pretty clever naming system.
It is pretty smart.
But I'm guessing this level of control over DNA, it didn't just happen overnight.
Oh, definitely not. The guide actually traces the history of oligonucleotides way back to the early days of molecular biology.
Like when exactly?
Well, the term molecular biology itself was coined in 1938, and that's around the same time scientists were beginning to understand even the very basics of how genes worked.
Oh, wow. So this has been a long journey.
Inez.
Of course, the discovery of DNA's double helix structure in 1953 by Watson and Crick.
Huge turning point.
That must have been a turning point, yeah.
Absolutely. Total game changer. That discovery allowed scientists to actually start thinking about creating DNA artificially.
Wow.
And the first attempts at actually synthesizing DNA strands happened just a couple of years later, in 1955.
So things moved pretty quickly after that initial breakthrough.
They did. There was a lot of excitement and a real push to figure out how to work with DNA in the lab.
But I'm guessing those early synthesis methods were pretty basic compared to what we can do now.
Oh, you're absolutely right. The guide specifically highlights the development of something called phosphoramidite chemistry by Marvin Carruthers in the early 1980s as a major leap forward.
Okay, so this was big.
This new method made it possible to produce large quantities of oligonucleotides with a high level of accuracy.
Wow, so before Carruthers came along, making these oligonucleotides was a lot more difficult and time-consuming.
Yeah, a lot. Carruthers' method was a huge improvement in terms of both efficiency and accuracy. It's kind of like going from carving a statue by hand to using a 3D printer.
That makes sense. So with these more advanced techniques, scientists can now create DNA in the lab that's basically identical to what's found in nature.
Exactly. And not only can they replicate existing DNA sequences, but they can design and synthesize completely new ones, tailoring them for specific purposes.
That's incredible. It's like having the power to write new genetic code.
It is. It's like having a molecular toolbox filled with custom-made genetic tools.
Speaking of tools, the guide mentions 3 main components that make up an oligonucleotide: a nucleotide base, a 5-carbon sugar, and a phosphate group.
Those are the key ingredients.
It sounds a bit like a chemistry lesson, but how do these pieces actually fit together?
Okay, think of it like this. The sugar and phosphate groups, they form the backbone, like the spine of the molecule, and the nucleotide bases are attached to that backbone. There are 4 main types of bases: adenine, thymine, guanine, and cytosine, and they act like the letters of the genetic code.
So the order of these bases is what actually carries the genetic information.
Exactly.
Like a sequence of letters forming words and sentences.
Precisely. The sequence of these bases determines the specific instructions that are encoded in the oligonucleotide.
Okay, that's starting to make sense. So how do scientists actually manufacture these incredibly precise structures? It sounds like something out of Star Trek.
It's pretty amazing, right? They're synthesized using a process called solid-phase chemical synthesis.
So can they really control the sequence down to the individual nucleotide?
They can. And that level of precision is crucial because even a single misplaced nucleotide can totally change the function of the oligonucleotide.
Wow. So you've got different lengths, and the order of those bases really matters. Does that mean there are also different types of oligonucleotides?
There are, and the guide talks about how they're categorized based on both length and function. We already talked about how the ogomer terminology tells us about the length, but the function is where things get even more interesting.
Okay, I'm all ears. What are some examples of these different types based on function?
Well, think of it like a toolbox. You have different tools for different jobs, right?
Right.
In the same way, you have different types of oligonucleotides for different applications in molecular biology.
Give me some examples. What kind of tools are we talking about?
One really common type is called a PCR primer.
PCR primer.
Yeah, these are short oligonucleotides that act as starting points for DNA replication during a process called polymerase chain reaction, or PCR. They're like the go signal for the molecular copying machine.
Okay, so if you want to make lots of copies of a specific DNA sequence, you need the right PCR primer.
Exactly. Without the right primers, the PCR process wouldn't know where to start copying. It'd be like trying to build a house without a foundation.
Got it. That makes sense. PCR primers are one tool in the oligonucleotide toolbox. What else is in there?
Another important type is called a probe.
A probe.
Yeah, probes are designed to bind to specific DNA or RNA sequences. They're kind of like a molecular detective searching for its target.
So they're like little homing beacons that can find a specific piece of genetic information.
That's a great way to put it. And this ability to target specific sequences is what makes probes so valuable for things like genetic testing and disease diagnosis.
Fascinating. It's like they're programmed to find a needle in a haystack of DNA.
Exactly. And the applications just keep expanding as scientists come up with new and creative ways to use these oligonucleotide tools.
This is all so interesting. I'm already starting to see just how powerful these little oligonucleotides can be.
And we've only just scratched the surface. There's a whole world of applications to explore, from revolutionary new therapies to the potential to store the entire world's data in DNA.
Okay, hold on. Did you just say storing data in DNA?
I did.
That sounds like something out of a science fiction movie.
It might sound like science fiction, but it's rapidly becoming a scientific reality.
I can't wait to hear more about that. But first, I have a question about something the guide mentioned. Trisaccharides and tetrasaccharides. Those sound like they belong in a chemistry textbook.
Well, they do have a very technical sound to them, you're right, but they're simply a different way of classifying oligonucleotides, specifically those made up of sugar units. Remember, oligonucleotides can be built with different types of building blocks.
So not just DNA and RNA, there are other kinds of oligonucleotides.
Exactly. Those sugar-based ones are called oligosaccharides, and they play important roles in various biological processes, from cell signaling to energy storage.
So it's like a whole other branch of the oligonucleotide family tree. Can you give me some examples of those?
Sure. One you might be familiar with is raffinose.
Raffinose?
Yeah, a trisaccharide found in beans and beets. It's actually made up of 3 sugar units: glucose, fructose, and galactose.
Interesting. So it's like a 3-sugar combo. What about tetrasaccharides?
Those are just oligosaccharides with 4 sugar units. One example is pistachios, which is found in a variety of beans and other legumes.
Okay, so these oligosaccharides sound like they're important, but are they as versatile as the DNA and RNA types we were talking about earlier?
They're incredibly versatile. Oligosaccharides are involved in a wide range of biological processes, from cell recognition and communication to nutrient absorption and immune responses.
Wow, so they're not just sitting around in beans, they're actually doing important jobs in our bodies.
Exactly. Yeah. And as scientists learn more about the roles they play, they're finding new ways to use them in everything from drug development to food science.
This is getting more and more fascinating by the minute. It's amazing to think that these simple chains of molecules can have such a huge impact on so many different areas of science.
It is. And we haven't even gotten to the really exciting stuff yet. We still need to dive into the amazing applications of oligonucleotides in areas like gene editing, disease treatment, and yes, even data storage.
Okay, I am definitely ready for more. Let's
take a break.
Let's take a break and come back to those incredible applications in part 2.
Sounds good. Welcome back to our deep dive into the fascinating world of oligonucleotides.
I'm ready to hear more about those incredible applications you mentioned. We left off talking about how versatile oligonucleotides are, and you teased some really exciting stuff, like using them in gene editing and even for storing data.
It is exciting stuff. But before we jump ahead,
Okay.
I want to circle back to a type of oligonucleotide we touched on earlier. Antisense oligonucleotides. Remember, these are designed to target specific messenger RNA molecules, or mRNA.
Right. You said they're like intercepting a message before it reaches its destination. Can you remind me how it works exactly?
Absolutely. It helps to picture the cell as a busy city
Okay.
with DNA as the central library holding all the genetic blueprints.
Okay.
When a specific protein needs to be made, a copy of the relevant blueprint is sent from the library to the construction site. That's the ribosome.
The ribosome.
In the form of mRNA.
So the mRNA is like the courier delivering the instructions for building a protein.
Precisely. And antisense oligonucleotides are like those secret agents who intercept the courier and prevent the message from being delivered.
Wow, that's a great analogy. So by blocking the mRNA, they stop the protein from being made.
Yep.
That's incredibly precise.
It is. And that precision is what makes antisense oligonucleotides so promising for treating diseases caused by faulty or overactive genes. You mentioned cancer earlier. Well, imagine being able to shut down the specific genes that are driving tumor growth.
That sounds like a game changer. The guy did mention that antisense oligonucleotides are already being used in clinical trials for cancer.
They are.
Are there any specific examples of how they're being used?
There are. One example that stands out is a drug called mipomersen.
Mipomersen.
Which is used to treat a rare genetic disorder called homozygous familial hypercholesterolemia.
Oh, wow. That's a mouthful.
It is a mouthful. But it basically means people with this disorder have extremely high levels of bad cholesterol, which puts them at a much higher risk for heart attacks and strokes.
That sounds serious. How does mipomersen help?
Well, mipomersen is an antisense oligonucleotide that targets the mRNA for a protein called apolipoprotein B, which is a major component of that bad cholesterol. By blocking the production of this protein, the drug can help lower cholesterol levels and reduce the risk of heart problems.
So instead of broadly targeting cells like traditional chemotherapy, these antisense oligonucleotides are going after the root cause of the problem.
Exactly. And that's just one example. Scientists are exploring the use of antisense oligonucleotides for a wide range of diseases, from infectious diseases to neurodegenerative disorders.
Wow.
It's a really exciting area of research.
It is. It feels like we're on the verge of a whole new era of medicine where we can treat diseases at the genetic level with incredible precision.
It's a paradigm shift. Instead of treating the symptoms, we're going after the underlying genetic causes.
Yeah.
It's really a testament to the power of understanding and manipulating the genetic code.
Okay, so antisense oligonucleotides are one way to manipulate gene expression. The guide also mentioned using oligonucleotides to actually track gene expression, kind of like seeing which lights are switched on in a cell.
Yeah.
Can you explain how that works?
Of course. Imagine trying to understand how the brain works.
Okay.
It's a complex organ with billions of neurons,
right,
each with its own unique pattern of gene expression. To study this, scientists can use special oligonucleotides that are tagged with fluorescent molecules.
Oh, okay.
These tagged oligonucleotides are designed to bind to specific mRNA molecules in the neuron.
So if a gene is active in producing mRNA, the tagged oligonucleotide will latch onto it and light up.
That's a great way to put it. By observing the pattern of fluorescence, researchers can see which genes are active in different parts of the brain.
Okay.
And how that activity changes in response to different stimuli or conditions.
That's incredible. It's like having a window into the brain's activity.
It is. And it's not just limited to the brain.
Right.
Scientists can use this technique to study gene expression in any type of cell, giving them insights into how cells function, how they communicate with each other, and how they respond to their environment.
Wow. So we can literally watch genes turning on and off in real time.
Pretty much. It's a powerful tool for understanding the intricate workings of cells and the complex processes that govern life.
Okay, I think I'm starting to grasp the amazing things we can do with oligonucleotides.
Yeah.
To understand and manipulate gene expression. But you also mentioned gene editing.
I did.
Isn't that a whole other level of complexity?
It is, and it's another area where oligonucleotides are playing a key role.
Yeah.
Have you heard of CRISPR technology?
Yes. It's that gene editing tool that's been making headlines.
Exactly.
It's like a molecular scalpel, right?
Exactly. CRISPR allows scientists to make precise cuts in DNA. And oligonucleotides are used to guide those molecular scissors to the exact spot where the cut needs to be made.
So they're like a GPS system for gene editing.
That's a great analogy. The oligonucleotide acts as a guide RNA, leading the CRISPR system to its target within the vast landscape of the genome.
That's incredible. It's like having the power to edit the blueprints of life. What are some of the ways CRISPR is being used with oligonucleotides?
The applications are vast. Scientists are using CRISPR to correct genetic defects, develop new therapies for diseases like cancer, and even engineer organisms with specific traits. For example, researchers are using CRISPR to modify crops, to make them more resistant to pests, or to increase their nutritional content.
Wow, we're talking about revolutionizing agriculture and medicine, but I imagine there are also ethical considerations with a technology this powerful, right?
Absolutely. CRISPR is a powerful tool.
Yeah.
And with great power comes great responsibility.
Of course.
There are important ethical discussions happening around the use of gene editing, especially when it comes to making changes to the human germline, which could be passed down to future generations.
Yeah.
We need to carefully consider the potential consequences of these technologies and ensure they're used ethically and responsibly.
It's a lot to think about, but the possibilities are truly mind-blowing.
They are.
Speaking of mind-blowing,
Yeah.
I'm still stuck on the idea of storing data in DNA.
Oh, yeah.
Can we dive into that now?
Let's do it. It might sound like science fiction,
Right.
but it's based on a simple concept.
Okay.
DNA is incredibly good at storing information.
Okay.
Think about it. All the instructions for building and maintaining a complex organism are encoded in its DNA.
That's true. But how do you go from digital data on a computer to storing it in DNA? It's not like you can just copy and paste.
You're right. It's not that simple.
Yeah.
Remember how we talked about the 4 nucleotide bases?
Yeah.
Adenine, thymine, guanine, and cytosine being like the letters of the genetic code?
Yeah.
Well, scientists can translate digital information, which is typically stored as a binary code of zeros and ones,
Right.
into this 4-letter DNA code.
So it's like creating a genetic version of our digital file.
Exactly. They can synthesize short strands of DNA or oligonucleotides with specific sequences that correspond to the encoded data.
So each oligonucleotide represents a little chunk of the digital file.
Precisely. And these oligonucleotides can be stored in tiny vials, much like a miniature library of genetic information.
Okay, that's pretty amazing. But how much data can you actually fit into DNA? Is it even practical for large-scale storage?
That's the really mind-blowing part.
Oh.
The storage capacity of DNA is absolutely enormous.
Wow.
The guide says that just 1 gram of DNA could theoretically hold a zettabyte of data.
Wait, a zettabyte? What even is a zettabyte?
It's a lot.
Oh, is that like a lot?
It's 1 billion terabytes.
Oh my God.
To put that into perspective,
Yeah.
all the data centers in the world combined are estimated to store only a few hundred zettabytes.
Okay, so we're talking about storing a mind-boggling amount of data in a tiny amount of space.
We are.
But how do you actually retrieve the information once it's stored in DNA? Is that even possible?
It is. Scientists can use DNA sequencing technology to read the sequence of the DNA and decode the information back into its original digital form.
Wow.
It's like having a molecular decoder ring.
That's incredible. It sounds like something straight out of a spy movie.
Yeah.
But is DNA data storage really practical? Is anyone actually using it?
It's still in its early stages,
Yeah.
but there are already some really exciting examples. Like, for instance, researchers at Microsoft have successfully stored and retrieved a digital archive in DNA that included over 100 books, a high-definition video, and even a database of crop variety information.
Wow, so it's not just a theoretical concept anymore. It's actually being used to store real data.
Exactly. And because DNA is so incredibly stable and can last for thousands of years under the right conditions,
Right.
it has the potential to revolutionize long-term data storage.
Wow.
Imagine preserving our cultural heritage, scientific knowledge, and even personal memories for future generations in the very molecule that carries the blueprint of life.
This is all so fascinating. I can't believe how much ground we've covered, from antisense oligonucleotides to gene editing to data storage, and it all comes back to these tiny strands of genetic material.
It is amazing, isn't it? Oligonucleotides are proving to be incredibly versatile tools, and we're only just beginning to explore their full potential.
I'm already feeling a little overwhelmed by all this information,
It's a lot to take in.
but I'm eager to hear more. What else is there to explore in the world of oligonucleotides? We've covered so much in this deep dive into oligonucleotides.
We have.
From manipulating gene expression to storing data in DNA,
Yeah.
it's clear these tiny molecules are having a huge impact on science and technology.
It's been quite a journey, hasn't it?
It really has.
And we've only just scratched the surface. There's still so much to learn and explore about the potential of oligonucleotides.
I'm excited to see what the future holds for this field.
Me too.
But before we wrap up,
Yeah.
I'm curious about something. We've talked about all the amazing things oligonucleotides can do.
Right.
But what about the potential downsides? Are there any risks or challenges associated with this technology?
That's a great question, and it's an important one to consider. Like any powerful tool, oligonucleotides can be used for good or for ill.
Right.
It all comes down to how we choose to develop and apply this technology.
So what are some of the potential risks we need to be aware of?
One concern is the potential for off-target effects.
Off-target effects.
Yeah, remember how we talked about the importance of precision when designing oligonucleotides?
Mhm.
Well, even with the most advanced techniques, there's always a chance that an oligonucleotide could bind to an unintended target,
Oh.
potentially leading to unforeseen consequences.
So it's like accidentally hitting the wrong key on a keyboard.
Exactly.
One small mistake can have a big impact.
And in the context of gene editing or therapeutic applications, those unintended consequences could be serious.
Yeah.
That's why researchers are constantly working to improve the specificity of oligonucleotides and to develop better methods for predicting and mitigating off-target effects.
It sounds like a lot of ongoing research and careful consideration is needed to ensure these technologies are used safely and effectively.
Absolutely. Beyond the scientific challenges, there are also ethical and societal implications to consider.
Okay, like what?
For example, as the cost of oligonucleotide synthesis and sequencing continues to decrease, we need to think about accessibility and equity. Will these technologies be available to everyone,
Right.
or will they only benefit those who can afford them?
That's a crucial point.
Yeah,
It's similar to the concerns around access to new and expensive medications.
exactly.
We don't want to create a situation where only the wealthy can benefit from these advancements.
Exactly. We need to ensure that the benefits of oligonucleotide technologies are distributed equitably and that everyone has the opportunity to access these potentially life-changing treatments or tools.
So access and equity are important considerations.
Mm.
Are there any other ethical concerns we should be thinking about?
Another important consideration is the potential for misuse.
Misuse.
As with any powerful technology, there's always the risk that someone could use oligonucleotides for malicious purposes.
Okay.
For example, could someone with harmful intentions design oligonucleotides that target specific genes in a way that causes harm?
That's a chilling thought. It's like something out of a science fiction thriller.
It's a possibility we can't ignore. But it's important to remember that the vast majority of scientists and researchers are driven by a desire to improve human health and well-being.
Yeah.
They're working hard to develop these technologies responsibly and to ensure they're used for good.
That's reassuring to hear.
Yeah.
It sounds like open dialogue and collaboration between scientists, policymakers, and the public are crucial for navigating these complex ethical issues.
Absolutely. We need to have open and honest conversations about the potential benefits and risks of oligonucleotide technologies, and we need to work together to develop clear guidelines and regulations that promote responsible innovation and protect against potential harm.
It's a big responsibility,
Mhm.
but it's an exciting challenge. It feels like we're standing at the cusp of a new era of scientific discovery and technological advancement,
Yeah,
and oligonucleotides are playing a starring role.
I couldn't agree more. It's an incredible time to be involved in this field, and I'm eager to see what the future holds.
Me too. It's been an incredible journey exploring the world of oligonucleotides with you. I feel like I've gained a whole new appreciation for these tiny but powerful molecules.
It's been my pleasure, and I hope our listeners have enjoyed this deep dive as well. Who knows, maybe we've even inspired some future oligonucleotide researchers out there.
That would be fantastic. And to our listeners, thank you for joining us on this fascinating exploration. We hope you've learned something new and that you'll continue to be curious about the amazing world of science and technology.
Until next time, keep exploring and stay curious.